So if you have any proteins, so how do you characterize them? So you have to characterize them for their concentration, their identity, their purity, their structure or sequence, size, shape or conformation, surface charge and activity. So we'll talk it now one by one. So first is concentration. So if you want to determine the concentration of proteins in your samples, then you can go for the protein assay. For example, Lorry's method or Kegelar's method. Next is by using quantitative amino acid analysis, UV spectroscopy or ELISA. If you want to determine the structure or sequence of the proteins, then you can go for the N and C termini, amino acid analysis, peptide mapping and sequencing, carbohydrate analysis, mass spectroscopy, and disulfide linkage assay. If you want to determine the size of the proteins, then you can go for the electrophoresis assay, gel permeation chromatography, field flow fractionation, ultrafiltration, and light scattering. Next, if you want to identify the proteins, then you go for the N terminal sequence, peptide mapping, or specific bioassays, isoelectric focusing, like that. If you want to determine the shape or conformation of the proteins, then go for the cir- circular dichroism, X-ray diffraction, FT infrared, NMR spectroscopy, Raman spectroscopy, epitope detection, and specific binding. If you want to determine how or at what purity these proteins are pure or real, so you go for the RPHPLC, gel permeation chromatography, SDS page, field flow fractionation, contaminant specific ELISA, multi antigenic ELISA, immunoblot, DNA assay, or LAL test, for example for pyrogen. If you want to determine the surface charge of the proteins, then go for the isoelectric focusing, chromatofocusing, or ion exchange chromatography. And the activity of the proteins is determined by the bioassays which are performed in vivo as well as in vitro, and specific binding assays.